Okay, write in your notes that another similarity with a bacterial cell is the way mitochondria divide, and that this process is called binary fission, like you see here in this picture. Over here on the right-hand side is binary fission. So the real question, of course, is what all this evidence tells us about the evolution of eukaryotic cells, right? So look at the similarities just between these two pictures. So here's a bacteria, here's a dividing mitochondria. So take a look at those and see if you can see some comparisons between the two. That's where we're going. Now, do all cells have the same number of mitochondria? What do you think? Write in your notes that the number of mitochondria in a eukaryotic cell is determined by how much energy that cell actually needs. Meaning, a muscle or nerve cell may need more energy to do what they do than, say, a blood cell does. Remember that a blood cell basically carries oxygen and carbon dioxide, whereas a muscle cell causes movement. And a nerve cell has to pass on nerve impulses. So in that situation, those cells would need more energy. They both need energy, but just different amounts for what they do. Now, some cells have one very large mitochondria, while others may have hundreds of smaller ones. Remember, since mitochondria have their own DNA and reproduce on their own, they can be different sizes according to their genomes and their genetic codes, right? Just a reminder that both plant and animal cells need energy, so it should come as no surprise that both types of cells contain mitochondria. So if you don't have that in your notes and feel like you need to slip it in there, do that now. By the way, can you tell which of these is plant and animal? And could you describe 3 or 4 differences between the two? Hopefully that's becoming second nature to you. But just in case, I've labeled them for you. So here you see that both plant and animal cells have mitochondria. And why do they have them? Because they both need energy to do what they do. Plant cells need them as well. Okay, so what about chloroplasts? Now we already know that chloroplasts are plant cell organelles where photosynthesis occurs. Again, if you don't have that in your notes, you should probably slip it in there now, but that should be fairly straightforward. Now these organelles are a class of plant structures called plastids. And there are 3 kinds of plastids to get in your notes. So let's get those in there now. Amyloplasts, you see the word right here, amyloplasts, are what store starch in roots and tubers. While chromoplasts, here's the word chromoplasts, chromoplasts are what store the plant pigments for fruits and flowers. However, it's the chloroplasts that store all the chemicals necessary for plants to convert carbon dioxide into sugar through photosynthesis. Now they're found in the leaves and other green structures of the plants and eukaryotic algae. Get that in your notes. Let's take a basic look at the structure of a chloroplast. First, you can see the actual cell wall right here. It's labeled for you. You can see the actual cell wall. So the cell that's housing now this chloroplast, that's its cell wall right there. And then in there now, you can see just barely on this side of it is the cell membrane as well. So remember, plants have a cell wall and they also have a cell membrane. Next, you can see the actual membrane now that surrounds the chloroplasts. All right, they call it the chloroplast envelope membrane. So it has its own membrane. That's very important. We're working towards now where these actual chloroplasts may have come from. Then just inside the chloroplast's own membrane is an area known as the stroma. All right, the stroma. And you see that label here, and it's all throughout the cell. Okay, so this is the stroma. Write in your notes that the stroma refers to the colorless fluid surrounding the grana within the chloroplast and is also the location of chloroplast DNA and chloroplast ribosomes. So get that in your notes. So this really acts like the cytosol, the stroma does. Remember, inside the cell right here is the cytosol. Well, the cytosol now inside here is called the stroma, but it basically does the same thing. It houses now the mitochondria's organelles. Now, we also see the thylakoid membranes. So here's the thylakoid membranes. There's my arrow. There it is. There's the thylakoid membranes. Get in your notes that a thylakoid is a membrane-bound compartment inside the chloroplast in cyanobacteria, and that they are the site of the light-dependent reactions of photosynthesis. So inside the thylakoids are where the light reactions occur. There's a light reaction and a dark reaction that we call the Calvin cycle, but that does not occur in the thylakoids, but the light reactions do. Now, lastly, this electron micrograph shows the grana stacks. So you see these grana stacks right here, okay? These grana stacks. Write in your notes that chloroplast thylakoids frequently form stacks of discs referred to as granum, or grana. The singular is granum. And we'll talk more about that and in greater detail in our unit on photosynthesis coming up soon. But that's the general structure now of a chloroplast. Okay, now, a typical plant cell might contain as many as 50 chloroplasts. Remember, we'll be delving into these organelles coming up in our next unit on photosynthesis and respiration. Now, the chloroplast is made up of 3 types of membranes we want to get in your notes. The outer membrane, you can see that here. There's the outer membrane. Now, this is freely permeable to all molecules. Freely permeable. Then there's a smooth inner membrane that you see here, which contains many transporters, which are integral membrane proteins. Now, you remember the function of a membrane protein is to get larger molecules from outside the chloroplast, or outside the cell in the case of our discussion, and into this chloroplast. Now, they would regulate small molecules like sugars and also proteins synthesized in the cytoplasm of the cell, but used now in the chloroplast. So those would be things that would move into the actual chloroplast. Then lastly, we just talked about this, chloroplasts have a disc-like structure called thylakoids. Now, thylakoids are where ATP is made. Remember, the thylakoids are where photosynthesis happens, and one of the byproducts of photosynthesis is the production of energy in the form of ATP. The question is, why in the world does it have so many internal membranes? The answer is that membranes increase the surface area for the membrane-bound enzymes that will eventually manufacture ATP to power photosynthesis and other cell functions. Make sure you get that in your notes. Now, again, we see the presence of membrane-bound enzymes now, and that the membrane looks exactly like the actual cell membrane structure of the plant that houses these chloroplasts. Remember, this is not the cell membrane. This is the chloroplast membrane. So, I mean, it looks exactly like the outside membrane of that cell. It's these membranes where photosynthesis takes place. And again, the mechanism of photosynthesis is one of our next topics in this class. But again, in your mind, you got to be saying, man, that looks exactly like the cell membrane that houses the chloroplast. Just like it looked exactly like the cell membrane that housed the mitochondria. Okay, so the point here is that chloroplasts can move and reproduce, which makes them semi-autonomous. And I want you to get that in your notes. Semi-autonomous. So because they can move and reproduce, it makes now them semi-autonomous. Autonomous makes, another word for it would be independent. So they're semi-independent because they can move, change shape, and divide, okay? And they can reproduce just like the mitochondria can because they have their own DNA. Now, all of these characteristics combine to make scientists believe that at one time, chloroplasts and mitochondria were their own separate bacteria. So these characteristics now that you see that we've listed lead scientists to believe, and they should be leading you to believe, okay, that these were once their own separate organism. You see the ribosomes in there? I mean, there's just too many similarities for that not to at least be a possibility. So let's review this theory of endosymbiosis and what sets mitochondria and chloroplasts apart from all organelles, and get these in your notes. All right, first, they grow and reproduce on their own. That's a huge one. Second, they have proteins that are manufactured by their own ribosomes, not the ones in the cell itself. Thirdly, they both have their own circular chromosome, which directs synthesis of their own proteins. And lastly, the organelles that they have are not part of the endomembrane system, which means they're not part of the cell's endomembrane system. They're part of their own endomembrane system. So that's really 5 really important characteristics, four really important characteristics that set mitochondria and chloroplasts apart from other organelles. So all of this information led to the formation of the endosymbiosis theory proposed by Lynn Margulis right here. You see her. Write in your notes that this theory proposed that mitochondria and chloroplasts were once free-living bacteria that were engulfed by an ancestral eukaryote and became what are called endosymbionts. You see that word right here. Endo means inside. Symbiont means somebody that shares now something with the cell. They both benefit is what it means. So they become endosymbiont, the cell that lives within another cell. Endo means within. Now, so in other words, they're their own cells that live within another cell called the host cell. Now, they both benefit from this living arrangement. One endosymbiont supplies the energy for the cell, while the other supplies food for energy, and in turn, the host cell supplies raw materials and protection for these endosymbionts. So make sure you get those characteristics in your notes. Okay, so that ends part 2. So now you have a better idea of where these chloroplasts and mitochondria may have come from. So take a picture of those notes, submit them to Moodle, grab that study guide and answer the questions, and we'll see you in the last section of our tour of the cell in part 3. Bye.